The second topic in Unit 2 is chemistry of life. What is this chemistry of life? It is a branch of chemistry where we learn about the molecules with which the living organisms are made of. So we learn about the chemical organization of all the living organisms. Through various studies of this chemical organization of living organism, it is understood that all the living all the biomolecules are similar to that of all the matters. That is, all biomolecules are made from small simple units called as the atoms, and atoms combine together to form the elements. So atom is the smallest particle with of an element, and all matters, including the living beings, all the living organisms including human beings, all matters are made of atoms. And atoms combine to form elements. If an element is made of a single kind of atom, then the substance is called as an element. Molecules are formed when two or more atoms bond together. If a molecule is made of one kind of atom, it is called as an element. If the molecule is made of two or more kind of atoms, then the substance is called as a compound. For example, here a hydrogen element, because it is made of two hydrogen atoms, but whereas when you take carbon dioxide, it has carbon and two different, that is oxygen. So here the substance is made of two different atoms, therefore it is called as a compound. Few examples for the elemental molecules. One is the hydrogen molecule, where it is made of two hydrogen atoms bonded together, and the sulfur molecules where 8 sulfur atoms are bonded together. So if you see in this substance, in the molecule, it is made of one kind of atom only. So it is called as an elemental molecule. Compounds, here it is substance which is made of two or more elements joined together by a chemical bond. One good example for a compound is a water molecule. If you take in the water molecule, you can see one oxygen atom bounded to two hydrogen atoms. So here this molecule is made of two types of atoms. So each molecule is made of one oxygen atom and two hydrogen atoms. So it is made of two different kind of atoms. So this molecule is called as a compound molecule. And good example of a biomolecule which is a compound molecule is a sugar molecule, where you can see one sugar molecule is made of 6 carbons, 12 hydrogen and 6 oxygen atoms. So there are 3 different atoms which combine to form this sugar compound. Yeah, learning about the basis of the biomolecules, that is all, they are all similar to all other matters made of atoms, elements and molecules. When we see what the living organisms are made of, what are the chemical molecules which make up a living organism, we see that 70% of the organism contains water. Water is a universal solvent, and therefore here the living organism also has water. 70% of it is made of water, since all the biochemical reactions occur in water. And next 1% is the inorganic ions like sodium, Cl minus, K plus, chlorine, potassium and phosphate. And small molecules like amino acids, sugars, nucleotides and fatty acids, they make around 6%, and the rest 23% is made of the macromolecules, big molecules, that is like nucleic acids, proteins, carbohydrates and lipids. So this is the chemical makeup of a living organism. And here we biologists, we are always interested to learn about the molecules. So here molecules, we know that it is a combination of different atoms. How are the atoms linked together is by forming linkages or bonds. So in this unit, we will learn about the biomacromolecules, but before that we will see how these macromolecules are formed. It is formed because of the bonds. So we will initially start learning about the different bonds found in the biomolecules, and in the later part of this lesson, we will learn about the different biomolecules. Chemical bonding. If you see the different biomolecules in the different biological reactions, we see there are different uh, interactions or chemical bonds being formed. So what are the different bonds found in nature are ionic interactions, covalent bond, and weak interactions, covalent interactions like hydrogen bond, hydrophobic interactions, van der Waals interactions, etc. We will talk in detail about these bonds. Here the ionic bond is formed when an atom when an atom wants to attain a stable configuration, it loses an electron to another atom, the other atom accepts it. So when one or more atom loses electron and the other atom gains them, for what is to produce a noble gas electron configuration and become a stable atom. So because of this donation and accepting electrons, an ionic bond is formed. So to attain a stable state, usually an atom with few electrons in the outer shell donates, and an atom with an outer shell that is almost complete usually accepts the electron. The atom that donates the electron will have more protons than the electrons, therefore they will get a positive charge. And these atoms are called as cations. The atoms that receive the electrons assume negative charge and they are called as anions. For example, in this case, let us take sodium chloride. We have an atom sodium where it donates one electron and the other atom chlorine accepts the electron. So they are now positively charged atoms here. Sodium donating the electron becomes positively charged, that is a cation, and the chlorine accepting an electron gains a negative charge, becoming an anion. These two oppositely charged ions are electrostatically attracted to each other to form the ionic bond, or it's also called as the polar bond. Covalent bond is one of the major chemical bond found in the molecules of living organism, and it is the strongest bond. It is found in all the energy rich molecules and all the essential molecules of the living organisms. How is this bond formed? Is by sharing of electrons in the outer orbital, atomic orbitals. Sharing of electrons between the atoms is it enables the atoms to hold together in the molecule. As I told you, it is the strongest and most stable bond. This picture represents the ionic comparative representation of ionic and the covalent bond. For ionic bond in the sodium chloride salt, so here sodium donates atom and becomes a positively charged cation, and chlorine accepts and becomes an anion. So because of this oppositely charged ions, they get attracted, forming the ionic bond. But whereas in the case of covalent bond, it is not donating or accepting but sharing of electron between the atoms. In a water molecule, if you take, you have an highly electronegative atom oxygen, and here the hydrogen atom to get a stable configuration, it is sharing its electron in the outer orbital and forming a covalent bond. So here sharing of electron forms a covalent bond between the atoms, which we can observe in a water molecule. Covalent bonds here, they can form double bonds. The atoms which forms the covalent bond, they can form single bonds, or you can also see double bonds formed between atoms, and these covalent bonds can be polar covalent bonds or non-polar covalent bonds. Now, polar covalent bonds are formed when there is an unequal sharing of electrons in the bond. For example, let us take in a water molecule, if you see the oxygen atom is a highly electronegative atom. So here the other atom sharing the electron is hydrogen. If you take here, this is a bigger atom, it is highly electronegative. So here the electron which is being shared by these two atoms, here it is unequal sharing, because this atom is a larger atom, and the sharing, it will be electron which is being shared will be more pulled towards the oxygen atom than the hydrogen atom. So there is an unequal sharing of electron, which leads to a polarity. Partial positive charge on the hydrogen and partial negative charge on the oxygen. So unequal sharing of electrons take place here, which results in a polar covalent bond. When there is an equivalent sharing of electrons in the bond, it leads to a non-polar covalent bond. In addition to the ionic bonds and the covalent bonds, we find various non-covalent interactions very essential for the stabilizing of the structures and for the molecular interactions between the biomolecules. So what are the non-covalent interactions we find in the biomolecules are, one is the hydrogen bonds, the second is the ionic interactions, third the van der Waals interactions, and hydrophobic interactions. These are all weak interactions and they are very short-lived, hence they are very essential for interaction between molecular interactions. So we will see in detail about these non-covalent interactions, what we find in biomolecules, starting with the hydrogen bond. If you see a hydrogen bond, it is then calling it as a strong bond, it is a molecular interaction which takes place between a hydrogen atom and a electronegative atom. So when does this interaction take place? If you see a hydrogen atom, okay, to attain a stable configuration, it forms a covalent bond with an electronegative atom like nitrogen or oxygen or fluorine. So here we saw in the polar covalent bond. Here this is a highly electronegative atom. So here when the shared electron is pulled more towards the larger atom, that is the electronegative bonding partner fluorine will pull the bonding electrons away from the hydrogen, and the hydrogen here will expose its protons, and the other side, that is on the unshielded surface, exposing its positive charge. So this positively charged surface is being attracted by another electronegatively charged atom, which is a part of another molecule, which we see here an interaction between two water molecules. So you can see here, that is in one water molecule, you can see a hydrogen atom covalently bonded to a oxygen. Because of an unequal sharing, the electron is pulled towards the oxygen atom. So the other side of the hydrogen atom is positively charged, which is having an interaction with, what is that? Interaction with the electronegative atom, which is a part of another molecule. So this interaction is called as the hydrogen interactions or hydrogen bonding. So most hydrogen bonds in biomolecules usually involves oxygen and nitrogen atoms. So here a hydrogen atom bonded to an electronegative atom, which is called as, usually we tell it as a donor, and the electronegative atom, which is involved in the hydrogen bonding, that is called as the hydrogen bond acceptor. So here keto groups keto groups, amine groups, imine, hydroxyl groups are usually hydrogen bond acceptors. They act as hydrogen bond acceptors, and you can see these in DNA, RNA, protein and complex carbohydrates. Hydroxyl groups, amine groups and imine groups are hydrogen bond donors. So they usually act as hydrogen bond donors. And when we see these hydrogen bond donors and acceptors, we can see that hydroxyl, amines and imines act as both donors and acceptors of the hydrogen bonds. So we know what is this hydrogen bond interaction, and here we can find the hydrogen bond in various biological systems, in genetic molecules and in DNA base pairing, in the DNA three-dimensional structure, and you can see in protein structure and also in the cellulose crystalline assemblies. Certain molecules or polyatomic ions resonate, that is, vibration or resonance is found in molecules or polyatomic ions. That is, the electrons are delocalized or they flow freely throughout the molecule. As the electrons fluctuate or flow through the molecule according to the natural resonant frequency of an atom, the dipole moment also fluctuates. The electrons are freely moving around the molecule. Therefore, the electron density will fluctuate in the molecule, leading to changes in the dipole moments. That is, what is the dipole moment? Is the electrical charges, separation of the electrical charges in the molecule. So there is a change. The dipole moment also changes. As the electrons flow, the dipole moment in the molecule also fluctuates. Therefore, all the molecules and atoms, they have an oscillating dipole. If you take this molecule, you can see at time 1, the one end is positively charged and the other end is negatively charged. But after a particular time 2, you can see this pole is now negatively charged and the opposite pole is positively charged. So the dipole has been changed, the charge has been changed at the poles. So here these molecules, in all the molecules which are close together, when these molecules are close together, the oscillating dipoles are sensed and they interact, they form weak interactions. So these weak interactions, they are called as this van der Waals interactions. So van der Waals interactions are weak interactions between oppositely charged regions by the nearby molecules. And this, it is very essential that the molecule should be very close. Only if the molecules are very close, these van der Waals interactions will take place. So these van der Waals interactions exist between any two molecules, and it is effective only at a very close range. And these interactions are due to the fluctuating induced dipoles. And it is the weakest of all the bonds. Though they are weakest, when it is combined, they are forming very strong interactions. And these van der Waals interaction can be formed between any two molecules. They play a very important role in intermolecular bonding between the biomolecules. For example, you can see in the protein structure, it helps in stabilizing the three-dimensional structure. You can see at this region, because of a that is, you have in the amino acid, there is a hydrophobic amino acid. So these hydrophobic amino acids, they can't interact with water. So they want to avoid interaction with water, and they are forming an hydrophobic interaction and they are clumping together. when they come close together, they are able to form, they are bonded by the van der Waals interactions and stabilizing the three-dimensional structure. As we saw, these van der Waals interactions are weak interactions, but here when they combined together, when it is a combined van der Waals interactions are found, they have a very strong strength. So here they create highly ordered complicated structures like proteins or any biopolymers. They form three-dimensional structures of proteins is stabilized with the help of this van der Waals interactions, and overall shape of any polymer is stabilized with the help of van der Waals interactions. And you can find van der Waals interactions in water molecules also and in individual sheets of graphite. And they also, this combined strength of van der Waals interaction helps in interaction you can between the surfaces. So you can see in geckos, they can walk steadily on a slippery surface because of van der Waals interaction between the surface and the microscopic hairs on the legs of these animals. The third type of non-covalent interactions are these hydrophobic interactions which you find in the biomolecules. This interaction arises when there is a contact of hydrophobic molecules with water. The hydrophobic molecules are usually non-polar molecules. They repel water. They cannot interact with water because they have long chains of carbon. So they will not be able to form hydrogen bonds with water. So what these molecules, hydrophobic molecules, do when they are dissolved in water, they cannot form hydrogen bonds, so they interact among themselves, clumping together to form forming the hydrophobic interactions. So clumping of these hydrophobic molecules together will allow them to have a minimal contact with water. This hydrophobic force also tends the water molecule tends this water molecule to form a cage structure around this hydrophobic interactions. And this hydrophobic interaction between the non-polar molecule is a very spontaneous process. If you see this hydrophobic interactions, we can see it has a very high importance in the biological structures. We can see in the cell membranes the phospholipids where it has a hydrophilic head and the phosphate group, hydrophilic head and the fatty acid chains, that is hydrophobic. So this helps in making the cell membrane, and in protein structure, in protein folding, the hydrophobic interactions play a very major role. And not only in that, you can also find in structures the hydrophobic surfaces as a protective coating. In case of leaves, you have a hydrophobic coating, and in the bird feathers also we can see the bird feathers have a hydrophobic coating. So hydrophobic interactions play a major role in structures and also in the molecular structures. We can see it plays a major role. Overall, all the non-covalent interactions, let it be the hydrogen bond or the van der Waals interactions or hydrophobic interactions, they have a very important role in various biochemical reactions and also in stabilizing the three-dimensional structure of the biomolecules. So here, if you take biomolecular interactions, they are all very highly specific. So this specificity is possible because of these non-covalent interactions. A specific reactions such as enzyme substrate reaction, antigen antibody reaction, and in signaling molecules between receptor and ligand. These are very specific reactions, and these specific reactions are possible because of these non-covalent interactions. And this non-covalent interactions also helps to stabilize the three-dimensional structure of the biomolecules like proteins and DNA. They are found in the biomembranes. All cells, they have an outer covering, that is their biomembranes, plasma membrane. This plasma membrane is formed because of the non-covalent interactions. If you take a cell, mostly non-covalent interactions are used in molecule to molecule interactions because it cannot use covalent. It doesn't prefer covalent bonds. Why? Because covalent bonds are more stable. So breaking, if you take in the interaction of molecules, it has to be quickly formed and it has to be broken. So that is not possible with the case of covalent bonds. Covalent bonds are very strong. It's very difficult to break and form, and it requires more energy. So nature, we can see it as beautifully designed that all the interactions, molecular interactions, are done with the help of this weak interactions, that is non-covalent interactions, because it can be rapidly formed and broken, that is associated and dissociated. So all the molecular interactions and functions depends on the non-covalent interactions.